Right. Hi, everyone. My name is Sarah Spendlove. I'm a fifth-year Bioinformatics PhD candidate in Dr. Valerie Arbeda's lab. I'm going to be presenting on a project that I've been working on for the last couple of years and under the advisement of both Dr. Arboleda and Dr. Sewell. And the title of my presentation is Polygenic Risk Score Analysis of Congenital Heart Disease Phenotypes, Elucidating the Contribution of Common Variants to Congenital Heart Defects. Congenital heart disease, or CHD, is a series of disorders where individuals are born with a heart defect. As you can see in this figure, only a small proportion of CHD cases have a known genetic cause. CHD can be caused by Mendelian mutations. It can be caused by aneuploidies like trisomy 21, but it can also occur sporadically. Penetrance of the disease also varies. So for example, if you take two people with the same known CHD mutation, they can have different types of CHD. And most CHD research to this point has been focused on rare variants. And studying rare diseases like CHD can be a little bit difficult because there are a lot less publicly available data sets, including whole GWAS analysis and whole genome sequence data sets. And the ones that do exist can be underpowered and unbalanced when occurring, especially the GWAS. And another interesting thing to note is that CHD includes a wide spectrum of heart or vascular defects. Also, can people hear me in the back? Okay, cool. So CHD has a wide spectrum. So some people have to have multiple surgeries as children and as babies, and some people just have a mild heart murmur that may or may not even be diagnosed. And so because of this, a lot of the less severe types of CHD aren't included in the CHD-specific GWAS, but they are captured by related endophenotypes like heart arrhythmia and heart murmurs. And our goal in this study was to investigate the genetic basis of congenital heart disease through the development of CHD-specific polygenic risk scores. And we hypothesized that these risk scores would give evidence that common variants contribute to both CHD risk as well as severity of CHD. For those of you who aren't familiar with polygenic risk scores, polygenic risk scores, or PRS, aggregate the effects of common variants across the genome. They are developed using GWAS as a base study from which the allele effect sizes are taken, so that for each individual in the target study, a weighted sum of the number of effect alleles included in each person can be created. So we use the base studies, specifically GWAS summary statistics, to generate the PRS model, and then the PRS model is used on the target data set to generate the individual level scores. And our target study is a collection of CHD whole genome sequences from the Gabriella Miller Kids First data set. We had access to 711 trios, 362 of which were of European genetic ancestry. And we focused on these ones since the GWAS summary statistics that we had available for CHD were focused in Europeans. And since we had trio data instead of case control data, we actually didn't have true controls since the probands get their risk alleles from their parents, except in the case of de novo variants. And to account for this, we used Plink to turn the untransmitted haplotypes from the parents into a pseudo control. And this allows us to be able to compare PRS in cases versus controls. All right, and the base study we are using are 3 CHD-related GWAS from the UK Biobank provided by the Neil Lab. And out of 5 phenotypes we looked at in our exploratory analysis, we settled on using the 3 phenotypes listed in black on this slide, which are abnormalities of heartbeat, heart arrhythmia, and heart valve problems or heart murmur. For clarity and ease, I will refer to these as heartbeat, heart arrhythmia, and heart valve in my slides. Note that the more severe CHD phenotypes listed in red were very underpowered and unbalanced due to the small number of cases, and so we didn't use them. So for comparison, UK Biobank has over 350,000 European controls, and you can see in these two GWAS, there's 427 cases and 313 cases. The GWAS results weren't very robust. As mentioned previously, though, CHD is a spectrum, and often people might just have a heart murmur. And so using endophenotypes is actually good at capturing both severe and less severe cases. And so those three that we're using are examples of endophenotypes. So using these three endophenotypes and two sets of variants, we developed 6 polygenic risk scores for CHD. Now, it's important to note that polygenic risk score studies usually validate the results in one of 3 ways to prevent overfitting. And you can see some documentation from Precise2, the PRS software I used, that explains the 3 main ways people usually go about validating the results. And one way is to use a validation data set, but we didn't have a genetically distinct set of whole genome sequences for CHD available. Like I said, with rare diseases, it's a little bit harder to find the data sets you need. Another option would be to split our target data into a test and a validation data set, but we already had to split our data to focus on individuals of European ancestry and didn't want to reduce our power further. And so we are using the third way to validate, which is to use permutation procedures to generate empirical p-values. And again, we calculated these using Precise2. PRS and ran them on our target data using a case-controlled disease model. We considered our empirical p-value significant if they were less than or equal to 0.05 adjusted by 6 multiple tests, or 0.00833. The empirical p-value listed is in the last column of the table. The original p-value before validation is in the second to last column, and the variance explained by the PRS is in the third to the last column. Note that this variance is based on the original p-value and not the empirical p-value, and so it is a bit inflated. The other columns in the table list the GWAS phenotype used, which variant list was used, the GWAS p-value threshold Precise used to pick variants from the base study, and the total number of SNVs included in the model. And you can see that both PRS generated using the heart valve phenotype are significant, and they stay significant once we validate using empirical p-values. And so those results are bolded in blue in the top of this table. Now, we wanted to look a little closer at this heart valve PRS using all SNVs. And so we generated some graphs. And we see here that the top decile of scores have an increased odds of CHD classification. And when we plot all of the PRS scores of cases and controls, we can also see how the mean PRS score is higher in cases compared to controls. Now, in addition to looking at CHD in a binary case control manner, we also thought it would be interesting to look at CHD severity. To do this, we assigned severity according to expert guidelines and gave each person a score based on whether they were pseudo-controls or had mild, moderate, or severe CHD. We then recomputed our 6 PRS and we generated the results seen in this table. Unlike in the case-controlled disease model, the heart valve phenotype produced the most significant results. Unlike in the case-controlled disease model, Only using fetal cardiac SNVs produced an empirical p-value that was significant after accounting for 6 multiple tests. But it is interesting to note that the heart valve PRS using all SNVs, so that's in black, bolded, as well as the heart arrhythmia PRS using fetal cardiac SNVs were significant before validation with empirical p-values, and so we count these results as nominally significant. We also made some graphs for these. In the Quantile graph, it shows that the change for each decile, and we don't see any significant differences like before in the other graph, but we do see that severity of CHD is associated with increasing heart valve PRS using fetal cardiac SNVs. And to show this in a different way, we plotted violin plots, and using Spearman's correlation, we calculated a PRS correlation with severity status of 0.136. To conclude, we found significant results using both a case-controlled disease model and a severity disease model. The GWAS phenotype that gave these significant results was the heart valve problem or heart murmur phenotype. This indicates its usefulness as an endophenotype for CHD. And we did not show the results here due to time, but we did look at these PRS in non-European trios that we had, but they were not easily transferable. And this is a known struggle with PRS studies, if you're familiar with it. And so future directions involve calculating non-European PRS for CHD. And I'd like to acknowledge both of my mentors, Dr. Valerie Arboleda and Dr. Jae-Hoon Sewell. There's lab pictures. And I'd like to acknowledge their labs for their support as well. And from those labs, I would like to specifically mention Leroy and Angela from the Arboleda lab, who helped with this manuscript, and also Darren, who was one of Jae-Hoon's big summer students, actually. And so they all contributed to varying degrees to this project. And the manuscript's not actually published yet, but will be on bioRxiv soon, so keep an eye out for it. And I'd like to thank QC Bio for their support and for giving me this opportunity, and acknowledge Kids First for providing the target data we used, the grants that have supported me, and the Bioinformatics IDP. Thank you. Any questions?